So we talked about what these concentrations look like, but this was all in the presence of ADH. So now let's talk about what it looks like when we do not have ADH. So where does ADH act? It acts at the distal end of the tubular system here. And so if we don't have ADH, we're not going to concentrate our urine as we go down here. So if that's the case, what is the concentration of the fluid going down here going to be? Well, you might think it's going to be 100 because there's 100 here. So it'll continue to be 100 because we're not removing any water. But actually you'd be wrong. If you were to measure the concentration of the fluid here in the total absence of ADH, you would actually see that the concentration was going down. So the kidney is actually further diluting the fluid in the collecting tubule. Now how does it do that? Well, you may remember that there are principal cells lining the collecting duct, and these principal cells have ATPases, which pump sodium out of the cell, and they also have ENaC channels. ENaC for epithelial sodium channel. ENaC channels that allow the sodium to diffuse into the principal cell, and so together these two allow you to reabsorb sodium. And that reabsorption of sodium is removing solutes from this fluid, and so it's making it more concentrated because the water cannot follow because there are no aquaporins because there's no ADH. So we see that these numbers have changed a lot when we don't have ADH, and is that the whole story? Well, unfortunately it's not. There's another little twist here. And that is that if you remember, this concentration of 1200 was made up of two things. One of them was sodium and chloride, which made up about 600 of the osmolarity. But the other thing was urea, which made up the other 600 milliosms per liter. And how did the concentration of urea here get so high? Well, it was because of this kind of circulation of urea, where urea just goes round and round and round and thereby increases its concentration. But the thing is that actually, it's ADH that makes the collecting tubule permeable to urea. So in the presence of ADH, you have urea channels here. And those urea channels are what enable the urea to get out of the collecting tubule and thereby circulate. So what does that mean? Well, that means that no ADH, no urea channels. And no urea channels, no recirculation of urea. No recirculation of urea, no high concentration of urea here in the medulla. You still have your sodium chloride concentration high, but you don't have a high urea concentration. And so actually, in the absence of ADH, your concentration in your medulla is not going to be 1200, but eventually it'll reach an equilibrium closer to 600. And of course, this 600 here, some part of that was also urea, so this is not going to be a full 600. It's going to be closer to maybe 450.